Hi, I'm the president and primary biochemist of Moondust Cosmetics. Welcome to this session of the Cancer Conference called Plastic Pollution, Climate Change, and Carcinogenesis, a Triad of Consequences. The preponderance of plastic pollution on land and sea has gained recent attention and is a huge ecological problem facing future generations. It has become common knowledge that plastics can break into microscopic particles or microplastics less than 5 millimeters in diameter, and the smallest may penetrate plant, animal, and human cells. Plastics or synthetics, which are not naturally occurring organic compounds and therefore not biodegradable, were a revolutionary discovery and have had many applications since their inception, from aeroplanes to spacesuits. The plastics industry for the manufacture of consumer products took off in the 1950s. However, since then, the worldwide production of plastics has been steadily increasing and replacing natural materials, even in the smallest items for daily use. Presently, the global production average is almost 400 million metric tons per year. It is estimated that 5 billion metric tons of plastics exist in landfills and in the environment and will degrade into tiny fragments or microplastics, which will migrate into living organisms. Traditionally, plastics are made from fossil fuels and are petroleum byproducts of the oil and gas industry. Vinyl chloride, or VC, is exclusively produced for polymerization into polyvinyl chloride, or PVC, a thermoplastic polymer that is widely used in the building and construction industry for pipes, doors and windows, wire and cable insulation, furniture, and medical devices. It is also employed in the packaging and transportation industries. PVC is the third most commonly produced synthetic polymer after polyethylene and polypropylene. Research into the environmental effects of plastics has revealed some alarming facts. The presence of plastics in seawater is likely to contribute to climate change by releasing greenhouse gases. Microplastics may also interfere with the biogeochemical carbon cycle of phytoplanktons, which absorb carbon dioxide from the atmosphere and replace it with the oxygen that we all breathe. Moreover, plastic pollution in the oceans may potentially contribute to species extinction, since seabirds are particularly vulnerable to ingesting floating plastic. However, birds are not the only species affected, as marine mammals, sea turtles, and humans, which all share trophic chain and habitat, have similar microplastics polymer profiles in their gastrointestinal and fecal samples, including polyethylene, polypropylene, ethylene propylene, polystyrene, and polyester. Cell culture studies are suggestive that microplastics can produce cytotoxicity, oxidative stress, induce immune responses, alter membrane integrity, and cause differential gene expression. All these cellular effects are associated with carcinogens at the molecular level. In 1959, internal industry experiments revealed micropathology in rabbit livers after repeat exposures to 200 ppm vinyl chloride monomer. In 1971, the first experimental evidence was provided that rats breathing VC vapors for 12 months developed tumors of the skin, lungs, and bones. In 1972, a further report showed liver angiosarcoma in VC-exposed rodents, and primary cancers of the liver and kidneys occurred at exposures as low as 250 ppm, while the allowable exposure limit for workers in the industry was 500 ppm. Later, VC administered orally or by inhalation to mice, rats, and hamsters produced tumors in the mammary gland. In the 1980s and 1990s, numerous other experiments established VC as a multi-site carcinogen in experimental animals. One notable experiment with mice demonstrated that chloroethylene oxide, a chemically reactive metabolite of vinyl chloride, by subcutaneous injection and in a skin application protocol, caused a massive increase of fibrosarcomas at the site of injection, or increased the incidence of squamous cell papillomas and carcinomas at the site of skin application. This is particularly significant because chloroethylene oxide is also a metabolite of vinyl chloride in humans. Route of administration in animal studies have included oral administration, inhalation, subcutaneous injection, intraperitoneal injection, and transplacental and perinatal exposure. Inhalation is the most significant form of VC exposure for humans since it is a gas. Metabolism of VC occurs in the liver subsequent to its absorption. Phase 1 metabolism occurs primarily via the cytochrome P450 isoenzyme and generates the reactive intermediates chloroethylene oxide, or CEO, and chloroacetylaldehyde, CAA. These are further metabolized in phase 2 reactions to end products, which are excreted. Both CEO and CAA can interact readily with DNA to form adducts and produce mutagenic effects, but CEO is the more reactive of the two species. Epidemiological data started to accumulate in parallel with laboratory animal experiments. In the 1970s, case studies appeared about rare liver cancers or angiosarcoma among VC and PVC production workers. As a result, the industry was forced to reduce time-weighted average workplace exposure levels from 500 ppm to 1 ppm by OSHA, the US governmental agency. Simultaneously, there was also increasing evidence of excess cancers in non-liver sites, including the brain, lymphohematopoietic and respiratory systems, and data was collected from larger epidemiological studies. Moreover, an increased lung cancer risk was reported in workers exposed to PVC dust. In 2008, a scientific review by the International Agency for Cancer Research, or IARC, concluded that there is sufficient evidence in humans for the carcinogenicity of vinyl chloride. Vinyl chloride causes angiosarcoma of the liver and hepatocellular carcinoma. There is sufficient evidence in experimental animals for the carcinogenicity of vinyl chloride. There is sufficient evidence in experimental animals for the carcinogenicity of chloroethylene oxide. However, IARC stated that there is contradictory evidence that exposure to vinyl chloride is associated with malignant neoplasms of connective and soft tissue and inconsistent or scanty evidence that it is associated with cancers of the lung, brain, lymphahematopoietic system, and breast, or with melanoma of the skin. The latter statement is rather surprising since both animal and epidemiological studies had provided significant evidence to the contrary. Previously, it has been proposed that a three-pronged approach may be used in identifying new carcinogens, including epidemiology, laboratory animal studies, and cell culture studies involving a statistically significant DNA damage and cell death apoptosis assay in a panel of cell cultures derived from different human tissue types to determine the carcinogenicity of a particular compound. Cell death is becoming an important parameter in cancer research as carcinogenesis is being increasingly connected with aberrantly regulated apoptotic cell death and the resulting deregulation of cell proliferation. Consequently, it is of paramount importance to reevaluate the current methods for cancer risk assessment in the case of vinyl chloride exposure within the context of an apoptotic model of carcinogenesis, and to consider a possible role for epigenetic mechanisms. According to this new model, subtle differences in gene expression in response to a particular carcinogen, genetic polymorphisms, can directly or indirectly initiate cell death and act as a trigger for two-step carcinogenesis by signaling an initial elevation in apoptosis or cell death, followed by a subsequent resistance to apoptosis and tumorigenesis due to a resultant imbalance in cell proliferation in target organs. Thus, more recent studies at the molecular level have revealed vinyl chloride-induced DNA damage in other human tissues which could be associated with cancer. In an interesting Chinese study, DNA damage to peripheral blood lymphocytes of vinyl chloride monomer or VCM exposed workers was measured. It was determined that certain genotypes involving single nucleotide polymorphisms in xenobiotic metabolism and DNA repair genes were associated with increased levels of DNA damage which signal cell death. In another Chinese study, the peripheral lymphocytes of occupationally exposed VCM workers were found to have a higher micronucleus frequency in a DNA damage assay than the control subjects. In addition, the level of chromosomal damage in the test subjects was higher in individuals with certain specific DNA repair polymorphisms in a related experiment. In a related experiment, micronucleus occurrence in Chinese VCM exposed workers was related to base excision repair polymorphisms. In fact, DNA lesions in the lymphocytes of occupationally VCM exposed individuals can include base pair substitutions, chromosomal aberrations, micronuclei, sister chromatid exchanges, and DNA strand breaks. Using the COMET assay, which includes the detection of DNA strand breaks resulting from apoptosis and other cell processes, the lymphocytes of VCM-exposed plastic workers showed significantly increased levels of DNA damage as compared with controls. The severity of DNA damage was correlated with duration of exposure, but not with age of the subjects. There is further evidence that certain genetic polymorphisms in p53 pathway genes may be associated with an increase in VC-induced chromosomal damage in plastic industry workers, and individuals carrying the variant genotypes are at higher risk for chromosomal damage than their wild-type counterparts. Such genetic differences could result in an elevated rate of apoptosis in the lymphocytes of susceptible subjects and represent the first step of carcinogenesis in an apoptotic model. Vinyl chloride carcinogenesis has also been associated with mutations in the p53 gene in the highly conserved regions of the DNA binding domain of the p53 protein, which is involved in the induction of apoptosis. Such mutations in p53 have been detected in human tumor tissue from liver angiosarcomas and hepatocellular sarcomas. In addition, a statistically significant trend of a mutant p53 biomarker has been detected in serum samples from vinyl chloride exposed workers as compared with non-exposed controls. Decrease in the activity of these mutant p53 proteins could represent the second step of carcinogenesis in an apoptotic model, conferring acquired resistance to apoptosis and whereby imbalanced cell proliferation would result in tumor genesis. Potential activation of epigenetic mechanisms that may be related to DNA damage and carcinogenesis have also been observed in the peripheral blood lymphocytes of VCM-exposed workers, involving the differential expression of specific microRNAs in subjects with a high micronucleus frequency. The epigenetic perturbations of some specific differentially methylated genes in peripheral blood lymphocytes of workers may also be associated with DNA damage from vinyl chloride. Moreover, mutations in KRAS2, an oncogene involved in cell cycle signaling, and methylation of P16, a tumor suppressor protein that plays an important role in cell cycle regulation, are frequently observed events in vinyl chloride-associated hepatocellular carcinomas. In addition, VCM exposure may alter telomere length, which could represent a potential biomarker of susceptibility to chromosomal damage. Exposed workers with increased micronucleus frequencies had significantly shorter relative telomere lengths than controls. Increasingly, adverse health effects have been observed in people living in the vicinity of petrochemical complexes. In one experiment, hepatotoxicity was caused by repeated and subchronic pulmonary exposure to low-level vinyl chloride in mice and involved an increase in caspase 3 and interleukin-6 levels in hepatic tissue. Interestingly, an elevation of caspase 3 and interleukin-6 was observed in the lung tissue of these mice following VC treatment for 3 weeks as well. Caspase 3 is an important mediator of apoptosis, and interleukin-6 is known to induce apoptosis in neutrophils. These results indicate that vinyl chloride may act as a carcinogen at lower levels of exposure than those experienced by plastic industry workers by stimulating apoptosis in specific tissues. In embryonic mouse brain, high-dose VCM inhibits the proliferation of neural cells and induces cell apoptosis, leading to an imbalance in the ratio of proliferation and apoptosis. In an experiment introducing nanoplastic particles into baby hamster kidney or BHK cells, a significant increase in the production of reactive oxygen species, which can stimulate apoptosis or cell death, was observed after 48 hours following PVC treatment. Flow cytometric analysis further revealed that the treated BHK cells were undergoing apoptosis in a dose and time-dependent manner. The above studies are indicative that vinyl chloride carcinogenesis can take place in tissues and organs other than the liver in animals and humans, since they are susceptible to VC-induced DNA damage and apoptosis in certain cases. These are most likely to include cancers of the lymphohematopoietic system, the lungs, and the brain. Although epidemiological studies seem to be scant, renal cell carcinoma is another convincing candidate due to the available collective data. In a large Canadian study, men were found to have a heightened risk for renal cell carcinoma associated with occupational exposure to vinyl chloride, and risk increased with duration of exposure. Animal experiments have shown that VC-exposed rodents develop primary cancers of the kidneys at relatively low exposures. Finally, treatment with PVC, which is composed of VC monomers, causes time and dose-dependent apoptosis in baby hamster kidney cell culture. Future experiments should include testing non-transformed human tissues for VC and PVC-induced apoptosis in lymphocytes, brain, liver, lung, and kidney cells to determine if there is the potential for a two-step apoptotic model of carcinogenesis in a tissue-specific manner. Additionally, some epidemiological data should be gathered on renal cancers associated with VC and PVC exposure in both men and women. Taken together with research on workers in the plastic industry, initial data suggest that the ubiquitous presence of plastics and microplastics in our modern environment, which can penetrate human cells and those of plants and other species, is a looming threat, potentially increasing the risk for a variety of cancers. Interestingly, vinyl chloride, a colorless gas with a sweet odor, has also appeared on a list of chemical weapons of warfare and terrorism that primarily injure the upper respiratory tract, including the lungs, and is a serious cause for concern in this era. In addition, evidence is accumulating that solid marine plastic pollution can contribute to climate change via various pathways and may be implicated in species extinction. So it is necessary to minimize plastic production while maximizing plastic recycling by increasing its current efficiency and eliminating deception by the industry. As a society, it is necessary to decrease our daily reliance on plastic and revert to natural alternatives like glass bottles, jars, and sackcloth, which have served us well in the past. Washing synthetic clothes is the single greatest source of microplastics in the environment, and a return to wearing clothes made from natural fibers could significantly reduce their presence. Cotton jeans with a cotton t-shirt are still an excellent choice. Hopefully, a decreasing reliance on fossil fuels will signal a decline in our love affair with plastics. Thanks for attending this session.